Hello everyone, today we are going to talk about embryology. We continue the topic of embryology, starting from the moment of implantation up to the end of the first month of development. Lots of interesting questions will be considered today, particularly the formation of twins, monozygotic, heterozygotic. Also we are going to mention the definition of chimeric organisms, and we'll find out that chimeras exist even between humans. And also lots of examples here from the database USMLE, from American database, because embryology is a very popular subject there. Okay, let's start, let's not waste our time. And last week we finished our lecture with the blastocyst, nearly the 7th day of embryonic development. It had its trophoblast, or the outer cell mass, and its embryoblast, or the inner cell mass. And exactly at this stage, inner cell mass, it segregates into two general groups: the epiblast, which is bluish in this diagram, and the hypoblast, which is yellowish. Notice that among the hypoblast, some cells are specified into anterior, visceral, endoderm. So the future cranial, or the future head region of the embryo, is already determined here during the moment of implantation. And this is a very important stage, because now those inner cell mass, those embryonic stem cells, if we take them out, as you remember, they were pluripotent. Now they are not equal, and among them we can distinguish different subtypes, and the process of the formation of the epiblast and the hypoblast we call delamination, and it takes place in the 7th day of the development. Later on, after the implantation, the embryo is looking like this. We can distinguish here the epiblast and the hypoblast, again bluish and yellowish, and some additional structures are formed. The amniotic cavity above the epiblast and the yolk sac cavity under the hypoblast. That's what you have to know. And in preparing for this lesson and in comprehending this topic, it is very important to imagine the visual patterns of the embryogenesis, because it is better to remember how it looks like and then to describe in words than to read the information in form of words. So the picture is very helpful in embryology. Okay, so let's look at this picture. The epiblast cells migrate upwards to form the roof of the amniotic cavity, while the hypoblast cells, they migrate downwards to enclose the yolk sac cavity. So 2 layers, 2 cavities, 2 vesicles, something like that. And trophoblast, we remember that this is the outer layer of the embryo, now splits into 2. The inner layer, let's look here, the inner layer is called cytotrophoblast. Cytos means cell, and trophos means nourishment. And in this way we highlight that this layer is made up of cells, distinct cells, while the outer layer, which is dark green in this diagram, it is represented by the syncytiotrophoblast. What it means: syn means common, cytio in this way cytos, cells, trophos again nutrition, and blast means that they divide. So syncytio means that cells are fused, opposite to cytotrophoblast, where cells, they have their borders, and with the lines you can see that cells, they are separated, they are distinct. And in the syncytiotrophoblast, cells, they something like fuse one with another with the formation of syncytium. And also in this syncytiotrophoblast, the characteristic feature is that spaces are formed. Those spaces are called lacuna, and on the 9th day of development they are still empty. So embryo is not in contact with the maternal blood. Still it gets nutrients only by digesting the tissues of endometrium. Okay, let's move on. And exactly at this stage we have to discuss the formation of twins. You know that twins could be different in the appearance, so dizygotic and monozygotic. In case of dizygotic, means 2 zygotes were created, two independent oocytes were fertilized. And it is important to understand that genetically they are like siblings, like brother and a sister, maybe two brothers or two sisters, and they differ one from another. They are not identical. Yeah, they're similar because they have the same mother and father, but they are different. And in this case, inside the uterus, we can find two chorions and two amnions, because they form two distinct organisms. Sometimes they might have their placenta fused, and due to those anastomosis between the bloodstream, they might exchange hematopoietic stem cells. And it is important to take into account, because after the birth, those people, when they are adult, they might have different stem cells, genetically different stem cells in their red bone marrow. It is important to take into account during the blood transfusion, and red bone marrow transplantation. Also, what is important to take into account in this case is that in case of different gender, if there is a boy and a girl, then male hormones might affect the development of the female organism, and in this case girls and even a woman along postnatal development might have some traits of the appearance characteristic for the influence of androgens, but in this case you should not suspect that girl itself produces lots of androgens. You should take into account that girl was affected by those hormones before the birth by brother's hormones. Okay, so in case you're going to be a pediatrician, you should take into account what was the character of the pregnancy, whether there were two siblings in the uterus, and so on. Okay, you understood. Also take into account that in humans, uterus destined to develop only one fetus. So in case of two or even more, we talk about pathology. And two twins, of course, is not a maximal number. There are many more could be there, and nowadays they could survive due to different technologies, modern technologies in neonatology. But this is not the subject of our lecture. Let's talk about monozygotic twins. They are genetically identical because they are formed from the same zygote. And depending on the stage when zygote decided to split, not zygote, but blastomeres, morula, or blastocyst, depending on the stage of development when embryo decided to split, we'll see different types of amnion and chorion. In case the fission of blastomeres occurred, let's look at this table, from 0 to the 4th day of development, we'll see two distinct fetuses. So they will have two chorions and two amnions, as in this case. In case, here they have different placenta. In case a fission occurred between the 4th and 8th week, days of development, then two distinct inner cell masses were formed, and in this case there is a common chorion but two different amnions. As you can see in the single chorion, single placenta, two different amnions are there. In case fission occurred between the 8th and 12th days of development, we'll find out that both embryos or fetuses later will have the same amnion, the same chorion, and it is the normal pregnancy. They could survive, and it's nothing harmful. But if fission occurs later, then in this case it will be conjoined twins, means that they could share their skin, some ventral organs, sometimes skeleton, and later this fission occurs, more harmful it is for the embryos, for the fetus. Okay, so what we have to say about monozygotic, I think it's obvious. They are genetically identical. Sometimes twins are born, much more if monozygotic twins are formed once in 7,000 of births, then 3 zygotic and more, 3, 4, 5 identical children are born much more rarely, but it is documented, it is possible it might occur as well. Okay, let's move on. And here we can see the example from the USMLE database questions highlighting the importance of the above mentioned information. So what is the question? Let's read thoroughly. Either fertilized oocyte cleaved in two on the day 6 after fertilization. Which of the following would best describe the pregnancy? And we can see among the examples dizygotic and monozygotic twins. In every case it is mentioned. And if we see that fertilized oocyte cleaved in two means that the same zygote gives rise to different fetuses. In this case we can exclude dizygotic twins, only monozygotic, yeah, because fertilized same oocyte splits into two. And day 6 after fertilization, if we return back, we see this table from 4th to 8th days of from fertilization. We'll find out 1 chorion and 2 amnions. This is exactly the case in this example. Okay, let's move on. And also I want to mention the information about chimeric organisms. They could be formed in animals and in humans as well. What is the chimera? Chimera, this is the single organism which is made up of several different, genetically different types of cells. How is it possible? In the laboratory it is possible when embryonic stem cells, so inner cell mass of one embryo, is injected into the morula of another embryo. For example, it could be the same organism, it could be mice and rat. So these could be stem cells from different species or from the same species, but it is important to take into account that the genome differs. They are not brother and a sister like that. Here, in this case, also embryonic stem cells or the cells of the inner cell mass could be injected into the blastocyst of another embryo. And in this case, the inner cell mass is formed out of 2 genetically different organisms. And this is exactly what is possible in humans if 2 blastocysts are formed, for example, in case of dizygotic twins, and in case one of them dies, but some cells survive and they could be absorbed by another blastocyst. Okay, imagine that. So in this case, one blastocyst could contain embryonic stem cells of 2 different, genetically different organisms, like 2 sisters or 2 brothers, or even a sister and a brother could be in the same organism. It is difficult to imagine, but it was revealed really some cases in humans when one woman, she needed the transplantation of a kidney, and the genetic analysis was performed of her kidneys and of her bloodstream and of all her relatives, and it was revealed that really her sons are not her sons. She is not so closely related to them, and it was possible because this woman itself was made up of 2 different cells, of hers and her sisters. Like the one organism was represented like by 2 sisters fused in the same organism, and her oocytes were produced by one cell while in her bloodstream and other cells were circulating. And that's really very interesting, and that's what you have to take into account. Okay, let's move on and return to our development. On the 11th and 12th day, we see the formation of the uteroplacental circulation. Starting from this stage, we can see that lacunae, you remember, they were formed in the syncytial trophoblastum. Now they are filled with the maternal blood. It is possible because embryo digests thin walls of maternal sinusoids present in the functional layer of the endometrium. So by breaking them down, embryo absorbs some blood from maternal bloodstream, and starting from this stage, it starts to grow much more intensely, comparing with the previous stages. Also, between the yolk sac and the chorion, the extraembryonic cavity appears, as well as the extraembryonic mesoderm, or the connective tissue put between the yolk sac wall and the chorion itself. Later on the 13th day, as you can see, the chorion cavity, or the extraembryonic cavity, the same, enlarges, and we can see that really embryo is put inside a large cavity of the chorion. Then the secondary or definitive yolk sac is formed by pinching off the exocoelomic cavity. This is the remnant of the primary yolk sac. So primary yolk sac is now split into two, the definitive and the exocoelomic cavity. Okay, let's move on. And now we are approaching to one of the most important information or definition in this lecture, the gastrulation. Gastrulation is the way in which gastrula is formed out of the blastocyst. So blastocyst, we have just previously mentioned what is it, becomes a gastrula. So now it contains the 3 germ layers. And in humans we have the key mechanism to create those 3 layers, and it is called immigration. We are going to discuss this a little bit later. Now please remember that it occurs nearly 14th, 15th day of development. So let's imagine how it is realized. It is better to imagine that there is the building with a 2 floors, and on the 2nd floor cells are put in the narrow room. This is the epiblast. And inside the epiblast, cells are very tightly packed because they increase in the number, but the space is restricted. And during the 14th day, in the midline of the embryo, in the midline of the 2nd floor, a line is formed. And this is the hole, linear hole, through which cells they start to fall down to the 1st floor. So if we look at the top of the epiblast, at the top of the embryo, here we can see the epiblast, hypoblast, and the top view is here. So amnion is removed to see better. And on the surface of the epiblast we see this groove. And this is exactly this place through which cells from the epiblast fall down and start to spread under the epiblast, forming first of all the endoderm. So cells falling from the 2nd floor and the 1st floor, they first get distributed around the 1st floor, and after then, when the continuous layer is formed, next cells migrating down, they form the middle layer between the 2nd and the 1st floor. The middle layer is formed, and this is the future mesoderm. So the process of falling down and their spread is called immigration. Immigration, because cells, they lose the intercellular junctions and they start to, to move. And this migratory phenotype is very important as it helps to form 3 layers. So through the process of immigration, cells of the epiblast, they give rise to all 3 germ layers. Notice that hypoblast, which is of yellow color, does not participate in the future development of the embryo itself. It gives rise to only yolk sac. Okay. Sorry. So epiblast through the primitive streak gives rise to all 3. Then cells remaining in the epiblast, they form the ectoderm. On this stage, it is important to mention that in the cranial or the head region of the embryo, so-called oropharyngeal membrane is formed. This is the place where epiblast and hypoblast, they are in tight contact one with another. Between them there is nothing, no mesoderm. And this membrane is the future oral cavity, ectoderm. We'll talk about it later. Okay, so just after the immigration, the process of immigration, we have to discuss how the notochord is formed. Notochord is the linear skeleton, the first body axis formed in the embryo. So once cells start to move through the primitive streak, besides at the top here of the primitive streak, so-called primitive node is formed, and cells from the primitive node are first to migrate downwards. So the first population of cells migrating through the primitive strict primitive node, they move forwards up to the oropharyngeal membrane, up to the precordial plate, and first they get intercalated into the hypoblast with the formation of the notochordal plate. So that's what is written here. So first population of cells, they're called pre-notochordal cells. They invade the primitive node and move forward cranially. Then, as it is written, they become intercalated in the hypoblast, and in this way they form the notochordal plate. Later on, they go into excisum and to form a distinct solid structure called notochord. Okay, they proliferate, detach, and form the definitive notochord. What is important about the notochord? Why we discuss its development? Because this is the first prominent structure formed in the embryo. It designates them the future axis, longitudinal axis of the body. And what is especially important, that notochord, it has an inductive effect, inducing the development of the neural tube above it. So all the cells of the ectoderm located above the notochord would give rise to the neural tube, and that's what we call the primary induction. So primary induction is when notochord stimulates overlying ectoderm to form neural tube, okay, to differentiate into neuroectoderm. Okay, let's move on. And now several genes regulating the body axis formation. So body axis, longitudinal, left to right, and dorsoventral. They are specified by several factors. Of course, this is very important. Of course, this is information only for 5. So if you are not interested, then skip, please. Then, first we have to talk about the anterior visceral endoderm, the anterior part of the hypoblast. It produces a special factor Cerberus, which inhibits the expression of different factors stimulating the formation of the primitive node. So in cranial region, the primitive node is not formed, and as you can see, the primitive streak doesn't reach the cranial end of the embryo. Then, in the dorsal end, in the caudal end, instead, the expression of Cerberus is low, so such factors appear and they stimulate the formation of a primitive node. So cells migrating inside and forming the notochord, they stimulate, they produce such factors to specify the notochord and the dorsal mesoderm. So under the effect of this, genes and their products. Actually, if something is written with the small letters, it means proteins. If with the caps letters, this means genes. Okay, so they're responsible for the notochord and dorsal mesoderm formation. Then remaining mesoderm, located more laterally, is specified by the bone morphogenic protein 4 and fibroblast growth factor as well. So in the embryo there is a gradient of concentration. In the center there is no bone morphogenic protein, but coming more laterally, its concentration increases, and it is important in the specification of the mesoderm into the dorsal, intermediate, and ventral. Okay, and also in the specification of the nervous system, we'll discuss later. What about the left-right symmetry? It is due to expression of several factors. For example, some factors like fibroblast growth factor 4 or zinc finger protein and also sonic hedgehog genes, they are expressed along the midline of the embryo, while the expression of nodal protein is shifted to the left side of the embryo due to high concentration of the serotonin. Serotonin, this is the 5-hydroxytryptamine. Okay, it is concentrated on the left side. Then also here we can see the increased concentration of such factors as lefty and eventually PTX2. This is the crucial factor in establishing left-right symmetry, because different problems with its expression result in the dextroposition of the heart, for example, or some problems with the normal location of internal organs. Also we had, I forgot to say, that guscoid gene is responsible for the formation of double-headed embryos in frogs and probably in humans. So this gene is also important to mention together with another set of genes. Okay, let's move on. And now information important even to get the minimal mark. Amnion. What is the amnion? And information about all the internal organs. Amnion is a transient organ providing the water environment of the embryo. It also absorbs the shock and facilitates the movement of the embryo. The wall of amnion consists of the epithelium derived from the epiblast and extraembryonic mesoderm. What is interesting is that amnion is the promising source of embryonic stem cell, of the fetal stem cells, because it has a very wide potential. As it derives from the epiblast, theoretically it could give rise to all derivatives of all three germ layers. So nowadays it is very popular to investigate its potential, the potential of stem cells of the amnion. Okay. Another extraembryonic organ which we have to discuss, this is the yolk sac. Yolk sac is formed from the endoderm. As you remember, we had the amnion and the yolk sac, two cavities. The yolk sac, it has also transient function as the source of nutrients. In humans, not very important, yeah, as we get almost all nutrients from maternal blood, from placenta, from the chorion. And it is more important as the source of the hematopoietic stem cells and primordial germ cells, future spermatozoa and oocytes. Notice that definitive hematopoietic cells are formed around the aorta at the level of the mesonephros. So we'll discuss this in the topic hematopoietic organs. But in any way, please notice, because all students, they say yolk sac is the source of nutrients. You have to remember about those two cell populations. And here is the example from the American database, and this question represents us the history of 1-year-old child with some problems, and doctors decided to prescribe the investigation with the help of technetium-99. This isotope is absorbed especially by the stomach epithelium, and in case of okay, let's first, I'm sorry, I just forgot to send you some information. So in the embryo, yolk sac is formed out of the mid-gut, the mid portion of the future digestive system, and sometimes we can find the yolk sac in the umbilical cord, but sometimes together with the yolk sac a part of intestine is involved, and so-called the Meckel diverticulum is formed. So because of the displacement of the intestines, as you can see, the small intestine get involved in the umbilical cord, this structure is formed, and usually in this structure the ectopic expression of stomach epithelium takes place. And to reveal this diverticulum, a special procedure is performed with the technetium-99, and normally it is absorbed by the stomach, and it is revealed in the left upper quadrant of the abdominal cavity, also in the suprapubic area, because urinary bladder might concentrate it. But if we can see the increased uptake of this isotope in the periumbilical region, this is the symptom of the Meckel diverticulum, and you should understand that it is formed out of the vitelline duct. So you see how important to know the embryology to diagnose different pathologies, even in adults, in this case in a child. So yolk sac could be the source of some abnormalities if it is if it involves intestines to the umbilical cord. Okay, now as for the allantois. Allantois is another transient structure, and in humans it forms cloaca, and it communicates cloacal region, urinary bladder, with the umbilical cord. So, as allantois is the communication between the urinary bladder and umbilical cord, in some infants, even in adults, we can see this pathology. Another example illustrates it. For example, here we can see that 1-week-old infant, it has yellow fluid leaking from his umbilicus. And the question is what embryonic structure is improperly developed or not degenerated in this case. And the correct answer is the median umbilical ligament, because exactly this ligament is made up of allantois, and under normal circumstances it becomes obliterated. So allantois communicates the urinary bladder with the umbilical cord, but under normal circumstances it obliterates, it collapses, it has no lumen at the moment of birth. But in case it persists, the urine might be leaking from the umbilicus in infants. So you have to understand why. You have to understand the function of the allantois. Okay, so allantois, it communicates a part of urinary bladder with the umbilical cord. Okay, then we approach into the next important process, the neurulation. Neurulation is the formation of the neural tube out of the neural plate. As we have already considered, the notochord induced overlying ectoderm to form the neural tube, exactly by inhibiting BMP4 expression locally. So if BMP is inhibited, then noggin, chordin, and follistatin, they stimulate the development of the neuroectoderm, while remaining ectoderm differentiates into skin. So BMP4 stimulates skin formation. In the central region, BMP4 is absent. So here, neural plate is formed. Then borders of this neural plate, they form grooves, they form folds, and those folds, they start to fuse in the midline of the embryo, and in this way the neural tube is formed. So neural plate forms the neural tube. Closure of the neural tubes starts from the cephalic region of the embryo and proceeds forwards and backwards. But before the complete formation of the neural tube, we can find the neuropores. Neuropores, these are the opening interior and caudal posterior openings before the closure. In the process of neurulation, very important role performs the folic acid. So folic acid is a vitamin, and it is often prescribed to pregnant women, because in case of its deficiency, multiple neural tube defects could be formed. But in case of folic acid treatment, those abnormalities are prevented. So remember, in also many MCQs, this question is asked about folic acid. You should associate with neural tube defects. Okay, but if we are talking about the derivatives of the ectoderm, we have to mention that it gives rise to the skin, neural crest, and neural tube. So, skin ectoderm, this bluish one, it gives rise to the epidermis and its derivatives like nails, hair, many others like sweat glands, sebaceous glands, like that. Also organs of oral cavity, because it invaginates inside the oral cavity and gives rise to teeth, tongue, cheeks, lips, epithelium up to the soft palate, and also adenohypophysis, because adenohypophysis is formed by invagination of the oral cavity ectoderm. Then neuroectoderm, exactly this neural plate gives rise to the nervous system, and development of nervous system is considered in another lecture. Then neural crest cell population is one of the most wonderful and interesting, as it migrates far from the neural tube. Originally, neural crest appears by each side of the neural tube by its closure. As you can see, neural folds, they get closed by each side. The neural crest cells are left, and here they form spinal ganglia or sensory ganglia here, and after that some of cells, they start to migrate, they colonize our skin, differentiating into melanocytes. Imagine all melanocytes, they arise from the neural crest, so they migrate far from the source of its origin. And also Schwann cells, odontoblasts, so cells of our teeth, for example, and many others. So this is the population participating in the formation of almost all the organs. For example, in our heart, it regulates the septation, so multiple heart defects, they result from the improper neural crest migration. And of course, it is important to mention that craniofacial structures like muscles, bones, and face development is due to the neural crest migration. Placodes, another example, and you have to know the placodes, these are the thickened region of the ectoderm, giving rise to the sensory organs. Here you can see an olfactory placode. Then this is the optic placode forming the lens, and also there is an otic placode. Otic means like future inner ear is formed. So placodes, they give rise to the olfactory epithelium, to the lens, and to the internal ear of the future human embryo. So we have considered derivatives of the ectoderm. Now let's talk about the derivatives of the mesoderm. Mesoderm is not a uniform layer, as we have already discussed. It has its dorsal part, which is programmed together with the notochord, you remember, and also intermediate and lateral plate mesoderm. So notochord, which is formed in the midline of the embryo, you remember that notochord has induced the neural tube to be formed. So the overlying ectoderm gives rise to the neural tube under the effect of factors produced by the notochord. Notochord itself gives rise to only intervertebral discs, so its input is ridiculous, but a structural input, but it has a very great influence and inductive role, inducing the most important system, nervous system. Then, by each side of the neural tube, there is a pair of somites or paraxial mesoderm. Also, it is called dorsal mesoderm. All terms are equally used, so it is better to know all of them. So somites, they belong to paraxial mesoderm or dorsal mesoderm. Dorsal because it is close to the back of the embryo. Paraxial means para-axis, like by each side of the central axis. Okay, so each somite is represented by three: sclerotome, myotome, and the dermatome. So it gives rise to bones, muscles, and dermis in the respective region of the body. So three different derivatives it has. Then, after the dorsal mesoderm, there is an intermediate put between the dorsal and ventral. Intermediate gives rise to the nephrotome. I'm sorry, this is another name. Intermediate means nephrotome, and it gives rise to the urinary and reproductive system or urogenital system. After that, the remaining portion is a lateral plate mesoderm. Lateral plate mesoderm splits into two leaflets, the parietal and visceral, and they give rise to the serosal lining. of peritoneal and thoracic cavity. For example, the lateral plate mesoderm gives rise to the parietal peritoneum, while the visceral layer of the lateral mesoderm gives rise to the visceral layer of the peritoneum. Also, what is important to mention that the visceral layer, or the splanchna pleura, gives rise to the heart muscle. Heart muscle in this way is a unique muscle derived from Rosen, from this splanchna pleura, or the visceral layer of the lateral plate mesoderm. So lateral plate mesoderm gives rise to serosal lining, and also do not forget that visceral layer gives rise also to the heart muscle. Okay, in this way you see that mesoderm gives rise to the wide spectrum of organs, particularly to the hematopoietic stem cells and vessels. Also they derive from the mesoderm. As for somites, due to the number of somites, or according to the number of somites, we can establish the age of the embryo. And here you can see the table, and you can see the respective stage and the number of somites. This is rather helpful in the early stages of the embryo. And then also here the diagram is put showing the inductive effects of different factors and how they specify the same somite to form three parts: the sclerotome, the dermatome, and the myotome. As you can see, the skin dermatome is specified by a special factor PAX3. Then PAX1 specifies the sclerotome, and some factors also are responsible for the myotome specification. It is not very important, so I'm not going to ask you about this during the practical lessons, only in case it is interesting. Okay, and here the USMLE example, which asks us that some researchers, they decided to investigate the limb development, and which of the following germ layers in the is implicated, which of the following germ layers is implicated in this experiment. So which of those layers is responsible for the limb formation? And as you know, mesoderm, it has sclerotome and the myotome, and limbs predominantly made up of skeletal tissue and muscle tissue. So among others, you choose the mesoderm in this case. Okay, and we approach into the end, and this slide demonstrates us the derivatives of the endoderm. So the lower layer is represented by the endoderm, and it is divided into the foregut, midgut, and the hindgut. Three parts, while the middle is communicated with the yolk sac. As you remember, that's what we were discussing earlier, when yolk sac sometimes involves a part of the intestine into the umbilical cord and stimulates the formation of the Meckel's diverticulum. Exactly here this abnormality might appear. So the digestive tube starts from the soft palate, starts from the pharynx, not from the oral cavity. Oral cavity organs, almost all derive from the ectoderm, but starting from the soft palate, the digestive tube is made up of endoderm up to cloacal membrane. Besides, oropharyngeal membrane is closed and it gets opened only on the 4th week of development, while cloacal membrane opens only on the 7th month of development. So much more later, and notice that digestive tube is closed on both ends early in embryogenesis. So what are the derivatives? Of course, all organs of digestive system from pharynx to the rectum, including liver, pancreas, gallbladder, as they are formed as outgrowth. Also it gives rise to the allantois, as it is shown here, and in this way allantois communicates the urinary bladder formed in the cloacal region with the umbilical cord, and that's what we have already discussed. So allantois might communicate the urinary bladder from the bottom belly. Then also respiratory system, as lung bud is formed as an outpouching of the esophagus, then almost whole respiratory system, starting from the larynx up to alveoli, is formed from the endoderm, and lots of endocrine glands are formed in the neck region from the pharynx. So in the pharynx, gills are not formed, but instead of gills, lots of glands like thyroid, parathyroid, C cells of the thyroid glands, and also tonsils and thymus. Many important structures are derived from the endoderm, particularly also I forgot to mention the eustachian tube communicating the middle ear and the middle ear itself with the inner lining of the tympanic membrane. They are also derived from the endoderm. Okay, and at last, the question from the USMLE database about the critical periods of development. The doctor is prescribing some drugs might be harmful for the embryonic development, and doctor insists or highlights the necessity to take contraceptives together with those pills. And the question is why a physician is concerned prescribing such drugs to the 26-year-old woman. The answer is between the 3rd and 8th week of development. I might confused you, yeah. So it might be confusing. Let's start from the beginning. So this 26-year-old woman of reproductive age, it is administered some retinoids. Retinoids, these are the derivatives of the vitamin A. This is the teratogen. It might cause different abnormalities of embryonic development. And antibiotics also are prescribing. So also the doctor said that this girl needs to take a pregnancy test before starting, because the pregnancy should be excluded before taking these pills. Why? Because those pills, they might have very harmful effect on the embryonic development. Okay, and the question is, during which period of development would physician be most concerned about the potential of a teratogen to cause severe damage that persists after birth? And we are aware that between the 3rd and 8th week of gestation, this is the period when general system of organs are formed, and exactly during this period, the effect of every teratogen is very harmful. So you should remember this term, and this is exactly the period we have considered during this lecture. And another question is about teratogen and teratogenic effect. Is when a 20-year-old woman she gave birth to an infant with a small head, low-set ears, widely spaced eyes, thin upper lip, and also some problems with the heart, probably the defect of the interarterial septum. So the question is to what teratogen the fetus was exposed to in the intrauterine period of development, and the answer is the ethanol, because the set of abnormalities is characteristic for this chemical. So it is very important to imagine that placenta doesn't provide a barrier between the mother's and blood and fetal blood. So lots of substances circulate and they are shared between the two. So especially if those substances, if those elements, pills or chemicals are taken by mother during the period from the 3rd to 8th week of development, they might cause severe abnormalities, and the spectrum of those abnormalities are characteristic for different types of teratogens, and you have to be aware about effects of all of them. I mean those 5 given in this case. And also it is important to highlight that it was for a long period of time it was thought that congenital abnormalities are due to mutations only, due to genetical defects or problems. But it was revealed that really different environmental factors, as well as medicines, many drugs, pills, and substances taken by maternal organism during the pregnancy are also very important, and they could affect the very significantly. So it should be taken into account, and the effects of those elements, they might cause different abnormalities, and several abnormalities are considered by the field of knowledge, which is called teratology. So teratology is the science investigating different abnormalities of development caused by different factors, environmental factors, while those factors causing such abnormalities, they are called teratogens. That's what you have to know, and what you have to differ from mutagens, because mutagens, they cause mutations, while teratogens, they cause abnormalities of development without changing the genome itself. Okay, this is the last slide of our lecture. Thank you for watching till the end, and I wish you good luck because the topic is very complicated. Please subscribe if you do really like our lectures, and put your thumbs up and put your comments below if you want to improve our lecture. Thank you.